Alright, welcome back to another lesson by Johnny Science. So, in this video, this student, well, he's having a bit of preparation for his exam now. So this is like a third occasion that we meet each other online in our lesson. So yesterday we did a lot of topics, like all the way until number 6 and 7. So 5, 4, 3, 2, 1, basically we haven't done that. Well, I just like to read things, backward, Now, so yesterday you described the first and the second Mendelian law of inheritance. But you didn't really explain the genetic linkage, crossing over, and things like that. Can you say that again? Can you describe X-linked inheritance, dihybrid cross, genetic linkage, and crossing over? Let's start with X-link.
Okay, X-link. X-linked inheritance refers to X-linkage.
Yes.
Linkage is the associations between genes on the same chromosomes.
Okay.
Therefore, the proportions of gametes were not shown, though expected by independent isosome.
So it basically X-link does not follow the second law of Mendel, right? Am I right?
Yep.
You have to
Yes.
say no, they don't. You have to say no, it doesn't follow. Not yet. Okay, remember. You have to say no, they don't. It doesn't follow. Okay. Otherwise, you will confuse the professors. Đây là cái sự khác biệt lớn giữa cái logic yes no của tiếng Việt và tiếng Anh á. This is the distinction between the yes no logic of Vietnamese and English. You have to answer no, it doesn't.
Okay.
All right, so continue with X-link, please. Please.
Continue with what?
So you said something about X-link. You said it doesn't follow the second law of Mendel, but how exactly? So the trait is expressed, or the trait follows only the sex chromosome. Am I right?
Yes.
Mm-hmm. So what does it mean then? For the boys and the girls, for the male and the female creatures getting affected. What does it mean then?
Sex linkage refer to carrying the genes on the sex chromosome, and it's different from the females and the males. Females have heterosome for sex chromosomes, while males are
both sex chromosome XX for female, while male only has
XY.
One X chromosome and one Y chromosome.
One Y chromosome.
You should also explain like the size of the X and the Y chromosome as well. The Y chromosome is much smaller, so it contains much less genes compared to the X chromosome. So what does it mean? It means that most of the traits, when most of the trait of X-linked inheritance is linked to the X chromosome, of course. Most, but of course there are some of the trait will be Y-linked as well, not just X-linked. So it means that the mother, mostly is the mother that determines the trait of the boys. However, for girls, how about that? She receives both X's from the mother and the father. So it's like her traits are determined by both of them, by both parents. But for the boys, it's mostly the mother. You have to understand that, link the ideas together. Phải liên kết các ý tưởng lại với nhau. Nó có lý do hết rồi nha. Nó phải liên kết lại với nhau như thế. You need to link ideas together in the right way, you know. Now, so can you give examples of X-linked inheritance?
X-linked inheritance examples: the red-green color blindness is a recessive condition. Yeah, as monocyte is dominant over it. So yeah, it's the recessive genes on the X chromosomes.
Okay.
X chromosome. So if the mother's is color blindness, it's 100% sure that all her boys is also color blindness as well.
Colorblind.
Colorblind.
Blind is an adjective, so blind itself is an adjective. So colorblind.
Colorblind, yeah.
Alright. I think it's better if you just try to write it down on papers in the exam and try to explain that. Now, so that's X-linked inheritance. Can you describe a dihybrid cross?
Dihybrid cross is the inheritance of two characteristic or traits. For example, when they'll study, it's bee plant. There are plants that have height and color which are more dominant than the short and color and white flower. The crossovers and receive the offspring receive the offspring tall and color plants.
So it means that what's the ratio?
The ratios for F1 is 100% of the dominance characteristic, while
of 100% of both dominance traits.
Dominant traits.
Or you can say it's 100% of both dominant phenotypes.
Well, in F2
generation.
F2 generations.
You need to say straightforward that F1 generation, F2 straight generation. Otherwise, F1, F2 can mean many things. You know. You have to say it like in full, like F1 generation, F2 generation compared to the parents' generation. You know, phải nói rõ ràng ra. Nó phải rõ ràng chính xác nhỉ. Một y học là phải nhảy. It has to be clear, precise, and concise as well. Phải chính xác và cô đọng. Xúc tích has to be clear, concise, and precise. That's how medicine is. Okay. Anything else? In F2 generation, yes.
Produce offspring with full phenotype.
That
which
follows the ratio of
full phenotype is 9-3-3-1.
Yes, 9-3-3-1. The follow ratio of 9-3-3-1. Okay, I think that's good enough. Now that's the second Mendelian law of inheritance. Okay, can you state the second Mendelian law of inheritance again? The law of independent assortment.
is the law of independent association when each pair of genes are differ from each others, and a pair of contrasted characteristic may combine with another pairs.
Okay, so basically the alleles, they segregate independently from each other according to this law, isn't it? Những cái alleles nó phân ly một cách độc lập so với nhau, nó không ảnh hưởng đến sự thể hiện tính trạng của nhau. So basically the alleles, they segregate independently. I get to say alleles segregate independently from each other. Then of course they do not impede or affect the expression of phenotypes of each other. They do not affect or they do not impede the expression of phenotypes of each other. They do not impede. Impede means like hinder, impede, or badly affected, negatively affect, impede, cản trở, prevent. Alright, so let's talk about genetic linkage and crossing over, maybe? Okay, let's start with genetic linkage first.
Genetic linkage.
Oh, by the way, have you done any kind of English tests and received your certificate before? Maybe IELTS, maybe TOEFL, maybe something like that.
Yeah, I did do IELTS.
Good. A long time ago, huh?
A year ago.
Okay, good. That's why your English is good. Compared to a lot of other students that I used to teach. Good. Now, so can you explain genetic linkage?
Genetic linkage. I thought it was the association between genes on the same chromosome.
Sorry?
I thought it is the association between genes and using genes on the same chromosome.
On the same locus, on the same chromosome. Locus, yeah. On the same locus.
On the same locus.
On the same chromosome. So basically, what does it mean? Some genes, they go together. So they don't separate, they don't segregate during like meiosis. And that phenomenon is called genetic linkage. So certain genes, they tend to stick together and they tend to segregate together, not from each other. They tend to go together. So they do not separate in meiosis, just like other kind of genes. So it means that if you have some trait 1, 2, 3, they might go all together in one creature. So it's got a phenomenon of genetic linkage. That explains why in the same family, certain traits will be preserved, in all the generations. Nó giải thích tại sao là trong một cái gia đình á, tại sao mà nhiều cái tính trạng á, nó lại có cái xu hướng là nó đi chung với nhau, đời con, đời cháu, đời chắc, y chang nhau luôn, với lại đời ba, đời ông, cha ông gì đó. So it explains that certain traits they go together and it is expressed in the generations, in many generations of a family, for example. That is genetic linkage. This phenomenon happens in animals as well as humans and plants as well. Can you give example of genetic linkage? This is hard.
I can write it, but I cannot.
Sure, you can write it on the chat if you want to. Or if you want, if you have like an iPad, we can share screen, and I'll show you how to share screen and write it.
No, I don't have an iPad.
Sorry?
I don't have an iPad.
Ah, never mind. Just write on the chat.
We have different traits that linkage each other. So in the next generation, F1 generations will create this genotype.
So it means alleles A and big A and alleles big B goes together. So it doesn't segregate. The same thing happens to alleles small a and small b, isn't it?
Yes.
Good. That's a good example. Now, let's talk about crossing over. Well, wait. Let's talk about the consequence, or the effects of having genetic linkage. It helps preserve certain traits in the same family. It helps preserve certain traits that goes together. It helps preserve certain things. Most of the traits that is preserved, I think, is good. That needs to be preserved. Otherwise, if it's bad traits, the creature dies before they have the chance to make babies, to reproduce. Nếu mà là tình trạng xấu thì cái creature đó nó sẽ chết trước khi nó có thể có khả năng mà gọi là tạo con của nó rồi. But so that's why I said that maybe most of the linked genes are good genes, of course. So it's preserved throughout the generation, which is a good thing. Now, so let's talk about crossing over.
Crossing over. It's a process.
It's a process.
Trading, exchanging alleles between homologous pairs of chromosomes in
homologous
in Prophase 1.
In Meiosis 1, yes, very good.
For it, it will have different gametes, produce different gametes. For example, I can write it down. For examples would be these parents. These parents can be exchanging alleles to turn into changing alleles and produce two gametes, that is large and small. Like this.
True. That can happen. However, you need to say that crossing over is very random.
Okay.
Crossing over is very random, it's spontaneous. It's a spontaneous process in which like genes are exchanged when the crossing over occurs between homologous pairs of chromosomes. And because of random pairing, it produces like an unlimited amount of combinations, một cái sự kết hợp mà gần như vô hạn, unlimited amount of random combinations. And what is the result of that?
The result is create a variations of
increase in the variation among the species, right?
Yes.
No, I think it's better to say it improves the diversity. It improves the diversity, or it increases the variation in the species, which helps them to, what does it do? Why do we need variety? Why do we need diversity? So when something bad happens, if most of the creatures die, some survivors that has mutations or mutants, or the genes that helps them to survive a catastrophe, they will continue to live on, and the creature will not become extinct. Một số cá thể khi nó sống sót một cái đại dịch hay là một cái hoạn nạn, một cái cơn thảm họa nào đó, thì là những cá thể còn lại đó nó sẽ giúp cái giống loài không bị tuyệt chủng, mà nó giữ cho cái giống loài đó nó vẫn tiếp tục sống. It keeps the creature, that kind of species, it keeps them alive and keep them alive and continue living, making babies. So that's why they need variations, need variety. Now, let's talk about cell division 1 and 2. Okay, can you describe what chromatin, chromosomes, and behavior of chromosome during mitosis?
Chromatins is the materials inside the nucleus when it's not coiled up, and chromosomes are when the chromatins is coiled up and getting more visible. Just when that called chromosomes. And chromosomes during mitosis consists of 4 phases: the prophase, metaphase, anaphase, and telophase.
Okay, and then another one. This is which is the longest phase? Is the interphase.
Interphase.
But interphase are basically not included in the mitosis. You should also mention interphase as well, Even though it's not included, you should also mention that, because it's the longest phase that the cell will span in.
Okay. During this phase, the centrioles migrate into different poles, opposite poles of the cells, and some of the microtubules is formed also form spindle fibers. The nucleus membrane disappears, and moving to the metaphase, the chromosomes arrange themselves at the equator of the cells.
Yes.
And the spindle fibers are
what is the spindle fiber? It's called mitotic spindle. Mitotic spindle. Nó có tên riêng, phải spindle fiber bình thường đó nha. spindle are basically fibers, same thing. This is a side spindles, but mitotic means it belongs to mitosis. So the name is mitotic spindles.
Spindle.
Where is it from? Where is it? Okay, the mitotic spindles are made of microtubules. Nó tạo thành bởi cái sợi microtubules. Mitotic spindles are made from microtubules, and it is made from the centrioles. And what does it do?
It helps. It attached to
the
centromere.
Mere means part. Central means the middle. Trung thể. Vậy rồi cái chỗ mà nó nằm, cái phần mà nằm ở giữa nó gọi là gì ta? Cái gì trung á, tâm động cái gì á? I don't know what the Vietnamese term is, but it's centromere. Yeah.
Centromere.
So the mitotic spindles are attached to the centromere of the chromosomes. What does it do next?
It's then pull to and separate.
It then pulls the chromosome to the two opposite poles of the cell. To the two opposite poles of the cell. Hai cái cực đối nghịch của tế bào. During anaphase, you have to say that.
During anaphase, yeah.
But okay, let's go back to metaphase now. During metaphase, you forgot to mention that the mitotic spindles line up the chromosome along the equatorial axis, isn't it? Nó line up, nó giống như là nó xếp hàng vậy đó, ở trên cái mặt phẳng xích đạo along the equatorial axis of the cell. I think you forgot to mention that part, very important part.
Em bảo là nó line up the equator of the cell.
Okay, you can say equator of the cell, true. Alright. And because they line up, so anaphase can happen easier and without much trouble. Because if they don't line up, when they move to the two opposite poles of the cells, they might, wiggle into each other. They might kind of coil into each other and causing problems, Nó sẽ cuộn lại với nhau, nó gây ra vấn đề đó. Nếu mà nó không line up, nó sẽ kéo về hai cực, nhưng mà nó kéo về cái kiểu gì ai biết được. Okay, so during anaphase, yeah.
During anaphase, the spindle fiber pull and separate the split and separate the chromosomes and pulling them toward opposite poles of the cells.
Opposite poles of the cell.
Yeah. Opposite poles of the cells. This process. This process is in need of energy, and so the energies are provided by the mitochondria around the spindle fiber.
Are you talking about like when the cells separate?
Yes, separate.
Okay, of course there's a formation of the furrow, cleavage furrow. Nó có cái tên riêng của nó là cleavage furrow. Cái sự tạo thành của cái cleavage furrow là cái chỗ mà nó tạo thành nó xoắn lại, nó tách cái cell ra. So basically, cleavage furrow is something that is formed during the end of telophase. Basically, it's just squeeze smaller, and it squeezes the two parts of the cells apart to produce two different cells. Alright, and and then the cell will enter interphase, which is the longest phase. You should also explain what happened during interphase. Well, G1S, G2 phase, you know?
Yeah, yes.
Of course, during interphase, all the materials are doubled. And what happens? The cell organs, the organelles of the organelles of the cell are also doubled. But you have to understand something. The two cells that are the result of mitosis, they do not necessarily have to have, like, you know, exact same number of organelles, exact same number of everything, you know, they don't necessarily have to have that. Nó không có nhất thiết, necessarily không có nhất thiết là sau khi mà mitosis diễn ra, hai cái tế bào con á, nó không nhất thiết là nó phải y chang số lượng organelles bào quang nha, không nhất thiết nha, nó có thể nó chia không đều cũng được nha, nó tùy vào cái nhu cầu của tế bào đó cơ. So basically it depends on the needs of the cells. After mitosis, the two resulting cells, the two daughter cells, they do not necessarily have to have the same amount of the same numbers of organelles like each other, but they must have the same number of chromosomes, the same composition of chromosome, you know, and genes. Nó bắt buộc phải có cùng cái số lượng chromosome, bắt buộc phải có cùng cái thành phần mà genes nữa. Mặc dù có số lượng bào quang nó không cần phải bằng nhau. Alright, what else? Okay, please describe G1S, G2 phase during interphase. You should also describe the checkpoint as well. Nó có một số cái điểm để mà nó kiểm tra checkpoint. Like for example, you have G0, you have G1S checkpoint, you have G2S checkpoint. You know, you remember them?
Cái checkpoint này để kiểm tra
Cái điểm
trước
để
khi
kiểm tra xem là, ví dụ như là G2S checkpoint là để kiểm tra xem là cái số lượng mà chromosome á, nó nhân lên nó có đúng hay chưa, nó kiểm tra lại xem có đúng hay chưa, nếu có vấn đề gì nó phải sót ngay lập tức, nên cái cell nó sẽ chuyển về G0 ngay lập tức để mà nó sửa sai. Còn nếu mà xong rồi thì nó mới tiếp tục được. Basically the checkpoint are there to check if the replication of chromosomes or DNA are correct or not. If there is a problem that needs to be fixed, the cell will undergo G0, which is like they stop everything and then try to fix the problem. After the problem is fixed, then the cell might go back into the interphase again and then continue on. Interphase can last for like a very long time because of many reasons. Because during the cell's life, interphase, like most of the cell's life is in interphase, you know. So it depends on the environment around the cells. If the environment is not favorable, nếu môi trường không có ưu đãi, không có ưu tiên, không có ưu đãi cho cái sự sinh sản, if the environment is not favorable for reproduction, then the cell will not reproduce. They will stay in interphase like a long time until the environment improves, cho nó khi môi trường nó cải thiện. Then, if the number of nutrients, the amount of nutrients increases, and if the signal for reproduction occurs, the signal can come from the cell itself, can come from the environment itself. Cái tín hiệu để cho sinh sản nó bao gồm là gì? Thức ăn dư thừa, dinh dưỡng, chất dinh dưỡng dư thừa, hoặc là cái tín hiệu đến từ cơ thể, hạn điều khiển cái tế bào, hoặc là cái tín hiệu đến từ cái tế bào bản thân nó luôn. If a signal for reproduction happens, then the cell will continue making new cells, like splitting. Now, mitosis does it happen to sexual cells, like for example eggs and sperm? Does it?
No, mitosis.
You should not say no. You should, because basically we are still finding new things every year. So this knowledge in the book, it's not 100% sure. So you should say that normally not. Thông thường, bình thường thì không. Chứ đừng nói là hoàn toàn không nha, không có gì khẳng định hoàn toàn 100% trong science đâu. Trong khoa học á. 10-20 năm sau người ta lại phát hiện ra cái gì đó mới chẳng hạn, thì kiến thức này sẽ cũ, nó sẽ bị lỗi thời. Remember, there's nothing totally 100% sure in science. So basically say probably not or normally not. So sperms and eggs normally do not undergo mitosis. Sperms and eggs are the result of meiosis. Nó là kết quả của meiosis đúng không? Okay, please describe. Okay, what is chromatin? What is chromatin?
Chromatin is the inheritance materials before get coiled up to become chromosome.
So basically, chromosome is the more condensed version of
chromatin.
Or maybe basically, chromatin and chromosome are basically
Same.
DNA. They are all DNAs, yeah.
Yeah.
But in different forms. Chromosome is a more condensed form. Nó là một cái dạng mà condensed hơn, đậm đặc hơn. And chromatin is when the chromosomes are not condensed. Okay. Now, can you describe meiosis and the raw meiosis in sexually reproducing organism?
Meiosis. Unlike mitosis, meiosis contains two meiosis, meiosis 1 and meiosis 2. As beginning of the meiosis 1 is occur interphase, which is the same to the mitosis.
Okay, meiosis 1 is basically similar to mitosis, except for 1 or 2 points. What's that?
The prophase and prophase 1. Prophase 1 and the metaphase. Prophase 1 is occur to have the homologous pair. Homologous pair up.
Homologous pairs.
Homologous pair up with another homologous pair to
so that crossing over can occur.
Yeah, crossing over can occur. And in the metaphase 1, this bivalence.
Formation of bivalence. Sự tạo thành của bivalence. The formation of bivalence during metaphase. So basically, during metaphase, it also randomly rearranged as well, isn't it?
Yes.
Nó cũng sắp xếp lại một cách ngẫu nhiên trong cái metaphase 1 nữa. Yes, very good. So 2 times. First is crossing over during prophase 1. The second is formation of the bivalence during metaphase 1. 2 cái lận. And they are both there to ensure the genetic variations. So that's cái sự mà đa dạng trù phú số lượng chủng loại về di truyền, genetic variation. Yes, please continue.
The spindle fibers attached to one side of the chromosomes. and separated this by valence by pulling the chromosome into different pole of the cells. Okay, pole of cells. And finally, in telophase 1, the chromosome arrive at the opposite poles and new nucleus membrane develop and form around a set of chromosomes on opposite poles, and spindle fibers break down, and in invagination occurs to separate into uh, two cells. And
so is there, the question is there interface between meiosis 1 and meiosis 2? Is there interface between 1 and 2 meiosis? Meiosis 1 and 2?
No, it doesn't have a...
There isn't. Khi mà mình nói không có cái gì đó, there is, there are nhé. Không phải là it has đâu. So there, when you want to say something exists, you say there is, there are. In the past, we say there was, there wasn't. Or there were, there weren't. In the future, you say there will be. Alright, chứ không phải là it has đâu nha. Là cái vấn đề lớn người Việt á, khi mà học tiếng Anh á. Okay, so this is a problem with Vietnamese people when they learn English. Earlier. Now, so there isn't an interface between meiosis 1 and meiosis 2. So what happened after telophase 1 then?
Telophase 1, after telophase 1, it went prophase 2 of the meiosis 2.
Does crossing over occur there?
No, crossing over that doesn't occur in the prophase
anymore, I see.
Prophase 2. Prophase 2 are the same as mitosis. Centrioles migrate to opposite poles. The chromosome line up at the equators of the cells. The spindle fiber attached to the centromere separate the chromosomes into and pulling them into different poles, and finally the chromatids arrived at different poles and uncoil new nucleus membranes.
Dissolve. The nuclear membrane dissolves, at the end.
Telophase 2.
So basically, during telophase 2, so basically, during meiosis 2, everything is very similar to mitosis, isn't it?
Yes.
True. Okay. So by the end,
it creates 4 daughter cells, which have half the chromosome of the parent cells.
The sister chromatids are now considered as chromosomes. Đổi tên á, có sự đổi tên. The sister chromatids bây giờ nó đổi tên, nó gọi nó là chromosome luôn. Right, okay, so good. So far, so good. Now, what is the role of meiosis?
The role of meiosis is to is its importance in genetic variation.
Yes. What else then?
What else?
It ensures the variation in the species, in the creature, in the species. So when there's a catastrophe happen, Can you please explain that?
The catastrophe happens.
When catastrophe. Or when
Catastrophe.
disaster happens, what happened?
Catastrophe that happens.
Some creatures will manage to survive.
Okay.
Some creature will manage to survive while the others die. And because some survivors are there, it keeps the species from being extinct. Nó giữ cái giống loài nó không bị tuyệt chủng. It keeps the species from being extinct. From becoming extinct. Alright. Let's move on to the structure of DNA and its role in heredity. The genetic code, the replication of genetic material. Cái này nó gọi là replication of genetic material, S-phase, you know, of interphase. Well, and then cái này là replication of genetic material chứ không phải là cái transcription và translation nha.
Okay.
So basically this is not transcription and translation. Basically we talk about replication of DNA only. Of course you will be asked transcription and translation too. Now please describe topic number 3. Let's start with structure of DNA then.
The DNAs are composed of nucleotides, which...
Okay, let me stand up then, I've been sitting. Okay. Nucleotides, right, did you say?
Nucleotides, and each nucleotide comprise of 3 parts: the phosphate groups, the pentose sugar, and the organic base. Organic base consists of
organic bases. The organic bases.
Bases.
Or we call it nitrogenous bases as well. Nitrogenous bases. Because basically they contain nitrogen.
Yeah.
Nitrogenous bases or organic bases. Different naming, you know. Okay, please continue.
Which are pyrimidines, which are 6 rings with 6 sides, which includes cytosins, thymines, and uracil.
Thymine.
Sorry, what? Thymine, yeah. And
purine, thymine, guanine, and adenine. Adenine, thymine, guanine, and cytosine.
I
think cytosine.
Okay. These three components give rise to nucleotide by condensation reactions.
What exactly is nucleotide? Nucleotide và nucleoside là hai thứ khác nhau nha. Be careful. Nucleotide and nucleoside are 2 different things. Nucleoside are only the sugar and the base. Nucleotide is basically the phosphate groups, the sugar, and the base. So be careful.
Nucleotides. A condensation reactions between the sugar and phosphate group or 2 nucleotides follow that allow the formations. We allow formations of polynucleotides. So creates a strand, and the DNA is a double-stranded polynucleotide arranged in double helix
In double helices, yeah.
and two strands entwined with one another's.
So the two strains are anti-parallel, or you can say anti-parallel. Anti is the same thing. Anti-parallel to each other. Song ngược chiều, anti-parallel to each other.
Around to each other. And coil and each coil measure about 3.4 nanometer. And there's these two parallel strands pairing the
Pairing to each other by using the nitrogenous bases.
okay.
And they are paired. They are paired with each other by the formation of hydrogen bonds between the nitrogenous bases or the organic bases.
Organic bases, where the orders the adenine and the thymines are linked together by two hydrogen bonds. And
sure, A and T by
two hydrogen bonds.
Two hydrogen bonds, and how about G and C?
C with three hydrogen bonds.
Right. This formation of hydrogen bondings, 2 or 3, it depends on the structure of the nitrogenous base. You have to also mention, like, basically, we have purine and pyrimidine. Basically, purines are A and G, isn't it? If I remember, purines are A and G.
Purine A and G, yes.
Yeah, pyrimidines are basically C, U, T. Yeah, C, U, and T. U are basically for RNA. In the case of RNA, we have U instead of like A. Anyway, because of the structure of the ring, so that's why it formed 2 or 3 hydrogen bonds. Okay, good. What else then? You forgot to mention that the chain, starting from the phosphate groups, it always goes from 5' to 3'. Từ 3 tới 5 phẩy đến 3 phẩy. It's like the chain always goes from the 5' to the 3' direction. And the opposite chain will go from, of course, it will go the opposite way. Okay. Okay, is that?
Yep, that's all.
Alright, that's all now. So, please describe the genetic code.
Genetic codes.
I think to describe genetic code, you have to describe like the transcription and translation process. And then you can describe the codons and anticodons, messenger RNA, tRNA, something like that. It's better to write them down, you know. It's better to write them down on papers and then you describe it, because when you describe like in words, sometimes it can be difficult for the professors to follow because they are older, you know. Remember, their ears are not that great like younger people. You become professors at the, say, minimum age of 30 years old. Minimum. But most professors I know, they become professors after like 36, 37, or maybe 40, something like that.
Genetic codes, like all the genetic codes is as all enzymes and are proteins, an organism characteristic can be determined by which enzyme are produced by the DNA. So different species organism possess different DNA, so it produces different enzymes, and due to the sequences of base pairings of the DNA
Determine.
determines the which proteins are manufactured. The sequences of amino acid in the protein determines the varieties of proteins, and there are about 20 types of amino acid, which uh, uh, which regularly occurs in proteins, and each should have
These
their
are
own.
just basically regularly occurring amino acids, but they're also something not regularly found because we are still finding new amino acids from time to time, you know. So they are unorthodox. They are like not conventional, unconventional types of amino acid, you know. Có những loại nó không chính thức, nó không chính thống. So remember, in science, there are always surprises waiting for you to discover it. So always be on the lookout. Never say never. Don't be too sure about your knowledge. Đừng có mà quá chắc chắn về kiến thức nhận được từ trong science. Nó không vậy đâu. Nó mỗi cái khoảng 10-20 năm người ta lại nghĩ người ta đã tìm được cái đó khác. After like 10 to 20 years, they will find out something new. So don't be too sure in science, okay? Okay.
With 4 different bases, it's possible to form about 64 different codes. And 64 different codes are triplet codes.
Triplets, or we call it codons.
Codons, yeah. And in the 64 codes, there are one nonsense codons, and 3 nonsense codons, and one start.
One start codon is AUG, and you don't say nonsense codon, we say stop codon, you know.
Stop codon.
We call them stop codon. There are 3 stop codon, which is UUG, UUG, UGU, right? Something U, I don't remember that last letter, I think.
UAA,
UUG, and then
no, UAG. They don't have UG.
UAG, and then what? And then U what?
UGA.
Alright, sorry. My memories are not so good now. UAA, UAG, UGA, yeah, these are stop codons. So what does it mean, these names then?
It means that when it reaches to the codon, the process of translations is stopped.
I mean, what does it mean AUG? Basically, okay, let's try to describe. Okay, so let's try to describe transcription and translation. This is because it's related to these things you just mentioned. Should also mention everything like this.
Transcription is the formation of mRNA using a portion of the DNA as a template. This
only one strain of DNA as a template. Một đoạn thôi nha. Well, after that, after everything, you should mention the replication of chromosome as well, which is a different process. cái số 3 này nó nhiều lắm nha, cái topic số 3 này nó nhiều thứ để nói lắm á. So there's a lot of things to talk about in topic number 3. Now, please describe transcription and translation. You need a break, or you need a break?
No, I'm fine. The DNA unwinds due to the breaking of hydrogen bonds between base because of the enzymes, and which leads to the DNA base being exposed, and its base with the help of the RNA primase, it creates primers at the start of the 5' of the templates. Begins complementing the 3' of the templates. Template DNA, and it begins to
Yes, what does it begin to?
Begin to add more complementary
bases.
Yeah. As more free complementary RNA nucleotides, and the strands keep extending as meets extending because of the I'm sorry.
Yes, because of that.
Along phase, free nucleotides expose base of DNA and move along the DNA. After transcriptions, the DNA close up and the helix form again.
The helices form again. And you should also mention the types of enzymes involved. Like, yeah, to open the helix and to close it, you need, what's it called? Helicase enzyme. Có enzyme đó nha, helicase đó nha. Helicase enzyme, and to stick the pieces together. You need ligase to stick the pieces of the DNA together, ligase. Nhưng mà để xoắn nó lại thì phải là helicase nha.
And transcription thì làm gì có helicase, ligase.
Wait a moment. Oh no, sorry. During the replication, during like synthesis of DNA replication, you have ligase. Yeah, true. Sorry. But during transcription, you need helicase to make the strand coil up again. You know.
Okay.
Okay. Very good. Very good. You remember right. Correct.
Okay. Formations. The DNA close up and the helix form against as the behind the RNA polymerase. And several mRNA are formed. And mRNA exists in nucleus. Through the nucleus hole and into the cytoplasms, and it went into the process of translations. Is involved the translations involve the formations of sequences of amino acids. That's code for
where this translation happens first. Okay.
The translation happens in the cytoplasm.
the cytoplasm? Are you sure?
Yes.
In the ribosomes, right?
Yeah, ribosome with ribosome.
We have ribosome floating freely in the cytoplasm. We also have ribosomes stuck on the endoplasmic reticulum. Which one does translation happen? Where does it happen? Which one?
Ribosome from the reticulum endoplasmic.
ER, very good, for the ER, very good. Now, okay, so please continue.
And it's involved with help of the transitions involve the rRNA.
Messenger RNA, yes. Translation involves messenger RNA and transfer RNA as well, tRNA.
So, the T first the groups of ribosomes, the subunit of ribosome attach to the mRNA and the tRNA carrying the amino acid with a complementary anticodons. It's attached to the first codons of mRNA and second tRNA amino acid complex with the complementary anticodon also attracted and attached to the second codon. That's the roles of the ribosome is to maintain the mRNA and tRNA complex, and this enables the process of forming peptide bonds between these amino acids. And when the amino acids is linked, the ribosome will move along the mRNA to attract more tRNA amino acids to attach to the codon of the mRNA.
Very good, very good.
Yeah.
So what happens next then? Once the stop codon reach
the ribosome detach. Ribosome.
The ribosome will stop the translation process.
Yes. And release the strand of polypeptide. That's all.
Release a strain of polypeptide. The polypeptide will have to undergo further post-translational modifications. Further post-translational modifications. Những cái sự mà biến đổi sau khi translation diễn ra nó gọi là post-translational modifications, you know. So it will become proteins, like useful proteins. So what happens with the messenger RNA after translation then? Simply floating in the cell now?
Sorry, can you repeat?
What happens to the messenger RNA after it has been translated? No information, right?
Yep.
There's no information mentioned in the book about what happens to that messenger RNA after it has done its job.
It disintegrates.
Well, probably it comes to the lysosome and gets dissolved. So you can have new materials for the next transcription happens and translation too. It's like a recycling process, you know.
Mhm.
Now, that is translation, transcription translation. Now, sometimes we do need to synthesize new DNA. Let's think about interphase, for example. You have G1S and G2 phase. So that S phase is when the DNA get replicated. Now, can you describe a replication of genetic material, which is replication of DNA?
Replications of DNA undergoes semi-conservative replications. Uh, it occurs during interphase uh, when the replications is controlled by the enzyme of the DNA, which includes helicase, the enzymes that help to separate and breaking down of hydrogen bonds. Also, a DNA ligase, uh, ligase,
Ligase.
ligase. Okay.
It's called ligase.
Okay. Uh, DNA replications includes following stuff: the enzymes, the helicase, force the DNA to adequate and and the hydrogens bond to break down so that the
It exposes.
exposes so
It
that's
exposes both strands of DNA.
so that the RNA primase can
Wait, RNA primase, right?
RNA primase, yeah, primase can you know
It's
can
weird. Why it has something to do with mRNA, right? Why it has something to do with RNA, but the name is RNA primase. Weird.
primase, yeah,
Black.
it's attached the primer, the primers to the beginnings the of the complementary strand, new strand, complementary strand DNA, and the DNA pool 3 help to uh, help to you know create new strand by adding complementary DNA nucleotide to the strand and move along until it uh,
Reaches.
reach the stop sequences.
Mhm.
Uh, and this replications process occurs in both strand
Process.
process in both strand of DNA, which leading to two type of the strands, which are leading strands and lacking strands.
Yes, and you talk about Okazaki fragments as well.
Yeah, okay, fragments. And okay, and, because of and at the end, it creates new, and this Okazaki fragment get attached to each other by uh, DNA pool one. Sorry, DNA pool one help to remove
Wait, DNA what?
DNA pool one
Polymerase 1.
polymerase one
Cái tên đầy đủ nó
help
là DNA Polymerase 1. Phải nói tên đầy đủ nha. DNA Polymerase 1. Then the full name is that. Which is the enzyme to do what?
which help to eliminate and cut off cut off primers the primers and with the help of ligase these strands these Okazaki fragments get to attach to each other to create a one single strand.
Okay.
Therefore,
Ligase will do that. Basically, Okazaki fragments and formation of leading strands are the opposite of each other, opposite direction. But they happen at the same time, isn't it?
yes.
Alright, now so happen at the same time, opposite direction, but then the Okazaki fragments will be linked together by ligase enzymes. And what happened after that? What happens after the leading strand and Okazaki fragment has been linked together?
The DNA separates to create two new DNA, which two new DNA contain one of the mother's DNA, one of the strand of the mother DNA.
Or the original DNA, I think it's better to say original DNA.
Original DNA.
Nguyên bản, nguyên gốc, original DNA. Okay. Now, let's talk about why do we need replication of DNA? Why is it even necessary? Why do we need DNA replication? Think about interface. Think about the fact that the cells need to double.
We need replications because it's necessary for for process of mitosis, which is process of which is very important for growing, repairing, and asexual reproduction. So it needs to replicate double replicated to create two daughter cells that have the same number of chromosomes as the mother.
It helps to double the amount of chromosome. True. Now, so my next question. So basically, translation and transcription translation ensure that the proteins for the cell's activities are made to make proteins. Yeah, am I right? Correct. Protein or polypeptides, if you want to make it that way. Meanwhile, DNA replication ensures that new DNA are produced to help the daughter cells to have the same number of DNA as the original cell. Isn't it?
Yes.
Okay, good. Now, number 2 is the most difficult topic you can ever talk. It's about glycolysis. Biological oxidation, it's what's it called? Respiration. Cellular respiration. Nó gọi là cái sự hô hấp tế bào. Cellular respiration. Basically biological oxidation, same thing. Photosynthesis. You need to talk about like pha sáng pha tối, what's it called? Light dependent. You need to talk about light dependent and light independent reactions. Is it phase or reaction?
Phase.
Thank you. Phases. And then basic characteristic of the enzyme, like Rubisco, for example. Sorry?
Yes, it's good.
Alright, and then photosynthesis also contains like electron transport chain, just like respiration.
NAD.
NAD, nicotine-amide dinucleotide, yeah. Nicotine-amine dinucleotide, yeah. Glycolysis as well. It's a long one. Glycolysis is like 10 steps. That happens in where does glycolysis happen? I forgot. Cytosol, right?
Cytoplasm.
Cytoplasm or cytosol is the same thing.
Yep.
Okay, and then Krebs cycle, where does it happen?
It happens in the matrix
of the
mitochondria.
And then later on, using these hydrogen created from using the hydrogen that is transported by the NAD. The NAD molecules transported the hydrogens to the membrane, outer membrane, okay. Màng ngoài mới đúng nha. Outer membrane of the mitochondria, and then the electron transport chain happens. You need to describe all these three processes. So Krebs cycle is also called citric acid cycle. It's basically the same, Krebs cycle, or tricyclic acid cycle, TCA. TCA, yeah, tricyclic acid cycle. Basically the same name. I mean, same thing, different names. All right. Biological oxidation, basically, I think it's electron transport chain, yeah. And Krebs cycle, because why? Glycolysis does not use oxygen, you know that, right?
Yes.
But then Krebs cycle and electron transport chain, you do need some oxygen. You do need a lot of oxygen, actually. And photosynthesis, well, you need oxygen. You need water, CO2. Alright, please describe glycolysis and biological oxidation first.
Sorry, can you repeat?
Can you describe glycolysis and biological oxidation first?
Biological oxidation. Only two process. Like the process of glycolysis includes 10 steps, which which occurs in 3 stages. First of all, the glucose gets phosphorylations, which phosphate group attached to both ends of the glucose. And moving into stage 2 is lysis, where the glucose diphosphates get breakdowns into three C molecules.
Three carbon
molecules. And the last stage is
what is that 3 carbon molecules? Pyruvate.
Oxidation. And the oxidation of these three carbons molecules to create two molecules of pyruvate.
Pyruvate, yes.
Yeah. Pyruvate. The pyruvate then undergo the linking phase to create
acetyl-CoA. Acetyl coenzyme A. It's actually acetyl coenzyme A.
Acetyl A, which is a two carbon molecules, and two molecules of acetyl coenzyme A undergo Krebs cycles inside the matrix of the mitochondria. At the beginning of the Krebs cycles, the acetyl-CoA gets to react with the ocellose acetate, which is a 4 carbon molecules to form citrates. Citrate.
To form what?
I'm sorry, what?
To form what?
To form citrates.
Citrate, very good.
Citrates. Citrates. And the citrates follow with the hydrogenations and decarboxylations.
The hydrogenation is the same as oxidation, you know. Loại bỏ hydrogen tức là em cộng vào oxygen. Tức là quá trình oxy hóa rồi. It's just the hydrogenation is just basically oxidation.
Yeah. And decarboxylations to create five carbon molecules and then 4 carbon molecules, and at the end
Oxalate, yeah.
The 4 carbon molecules get to like rearrange to become Can you spell the word for me?
Oxalate, you mean? Oxalate, I think. Let me check this one. Oxalate, I think. I'm not so sure if I remember. It's like oxalate something. I'm not so sure if it's a real name. Maybe I'm confused. Let me check. Sorry, oxaloacetate. Sorry, oxaloacetate. But I think by this point, if you mention these names, it's already too much. It's already good enough. Okay, good. Please continue.
And at the end process it creates.
At the end of the process.
The process it creates three NADH and 2 N molecules and 1 FADH2 molecules, and also create 6 carbon dioxides.
6 CO2 being produced, and then it's a waste product get out. Yeah. Yes, please continue.
And it undergo oxidative phosphorylation. Is the process.
Okay, you need to say what undergoes phosphorylation. Basically, it's the hydrogen collected from glycolysis, and during Krebs cycle, it's the hydrogen that being transported to the outer membrane of the mitochondria that will trigger the biological oxidation. Sorry, will trigger the phosphorylation something. Chứ cái phân tử glucose lúc ban đầu á, thực sự bản chất nó không có đi vô cái chuyện mà electron transport chain hay là phosphorylation gì đâu. Nó chính là cái hydrogen lấy từ cái glucose đó qua quá trình mà glycolysis và cái Krebs cycle, nó mới đi vào cái electron transport chain và phosphorylation gì nha. Phải hiểu được như vậy. Chứ bản chất cái phân tử glucose ban đầu nó không liên quan gì đến cái cuối cùng đó. Nó liên quan gì đến cái phosphorylation với lại cái electron transport chain đâu. So I was talking about that. Basically, the original glucose molecule at the beginning of glycolysis does not have anything to do with the phosphorylation and electron transport chain. It is the hydrogen.
Okay. Nhưng mà tại sao lại outermost tưởng là inner? Cái quá trình cái chuỗi vận chuyển electron ở trong màng trong.
Yeah, what's your question then?
Then tại sao anh bảo là outermost travel to the outermost membrane?
Thì nó mới ra cái vòng mà nó mới đi vào thông qua cái ATP synthase, nó mới tràn vào được chứ.
Vâng, phòng ra thì cái đấy gọi là khoang trung gian rồi.
Okay, fine, you can just you say that. But it has to travel through the ATP synthase, okay?
Yep.
And then it drives that ATP synthase to produce ATP. Okay.
The hydrogens carriers, the NAD and FAD carry the hydrogens atoms released from the glycolysis and the Krebs cycles to the outer membranes of the mitochondrion. The NADH and FADS then pass the hydrogens atoms that carries down in energy gradients, creates the release of transfers of hydrogen atoms, the energies, the release energies used to form ATP, which when FADH release its hydrogens to ATP are formed, while NADH release is create 3 ATP molecules.
That's a bit too complicated. But anyway, maximum you can produce like 32 ATP, I think. It depends on the amount of oxygen that is present. You know what, electron transport chain is really difficult. It's like confusing. Because some books they say this, some books they say that. Very, very confusing part. Okay, please continue.
As the carrier receive hydrogen atoms, these atoms split into proton and electrons, and only the electrons are transported in the electronic transfer in the electron transport change, and at the end of the change of the system, oxygen is the final acceptors of the electrons to form water. Yeah, and at the end of all the process, maximum is 36, maximum of 34 ATP.
32, 34 ATP, yeah.
34 are formed.
Oh, I think you forgot to mention the fact that glycolysis, it uses up 2 ATP and then produces 4, so the net is plus 2 ATP for glycolysis. Đừng quên cái vụ đó nha. Nó phải chi tiêu là 2 ATP cho cái glycolysis, nhưng nó tạo ra 4 cái cho nên net là cộng 2 ATP.
It's still 34 overall.
Yeah.
So that's the end of the...
Okay, good. Alright, let's talk about photosynthesis, which is of course happening in plants, algae, anything that has green substances. Oh, and you need to talk about the, what's it called, photon centers or something like that. Chlorophyll substances, chlorophyll something. This is the most annoying part of the whole study process. Photosynthesis.
Photosynthesis is
Cái
divided.
phần mà nó mệt mỏi phiền muộn nhất luôn á trong quá trình học sinh học á. It's like
Okay. Photosynthesis divide into two phases, the light dependent phase and light independent phase. The energies first start with the light dependent phase. After the light is trapped into the chlorophyll chlorophyll pigments, the release high energy electrons are released into photosystem, which includes pH 1 and pH 2. This
Photosystem 1 and photosystem 2.
to
Let's not use like abbreviation too much, okay? Đừng dùng cái viết tắt quá nhiều nha. Let's just say photosystem 1 and photosystem 2, okay?
two. Okay. These electrons are received by electron acceptors and pass along the electron carrier. ATP is generated from ADP, and this process is known as photosphorylation.
Photosphorylation. Wait, but first thing first. Cái đầu tiên nhất nó phải là cái quá trình mà khử nước nhỉ? Photolysis. The first thing first, you have to talk about photolysis, right? Which is the breakdown of water molecules.
Yes.
Using the energy of the sunlight.
Okay. There are two pathways of chlorophyll electrons travel, which are cyclic photophosphorylations and non-cyclic photophosphorylations. The electrons released from the excited chlorophylls combine with the electron acceptors and pass down the chains that end with NADH and NADP. The electrons are then returned to the electrons carrier system and recycle down the concentration gradient, thereby yielding ATP from ADP. And for the non-cyclic phosphorylation, the electron released from the excited chlorophylls combined with the electron acceptors pass down the change to the photosystem 1. The loss of those electrons are formed replaced by splitting waters. The process
by splitting water, yeah. And oxygen is a byproduct of that splitting of water.
Yeah. With other reactions, the protons help to form reduce a NADP, which is NADPH. Okay. DPH, and all of these materials, ATP and NADPH, then is necessary for the light independent process.
Light independent process, yes.
Process. The states involve dark reactions, which means it's occur regardless of
it occurs where? Dark reaction, where does it happen?
I'm sorry, what?
Dark reaction, where does it happen? Or light independent reaction?
It occurs regardless of whether light is presented or not.
Exactly. It just happened in the chloroplast, but it happens when the weather is dark. chứ nó không phải là diễn ra trong chỗ nào khác tế bào đâu. It's not, it doesn't happen anywhere else. No. And so it's happened when it's dark, when the weather is like not sunny, when it's dark. Yeah, not anywhere else in the cell, but chloroplast.
It occurs a process of called the Calvin cycle, and it occurs in the stroma of the sites.
Of the what?
The chloroplast. The pathway begins with the carbon dioxide fixations by a 5 carbon molecules called ribulose for biphosphates.
Ribulose. It's called ribulose 5-phosphate.
5-phosphate.
It's called ribulose 5-phosphate.
The product is a unstable 6 carbon compound that get to break down down to 3 carbon sugars called glyceride phosphates.
Called what?
Glyceride phosphates.
What?
Glyceride.
Can you write it down?
Glyceride.
Glycerate, you mean?
Glyceride.
Glycerate 3-phosphate?
Yeah, 3-phosphate.
Glycerate 3-phosphate.
Then the reaction is followed by 2 phases: the reduction phase and the regeneration of ribulose 5-phosphate. The reduction phase is the glyceride phosphates reduced to another 3 carbon sugar called triose.
Triose means like 3 carbon sugar.
Yeah.
Nó là cái thành phần mà nó có 3 cái carbon, nó có cái đường có 3 carbon, gọi là triose.
Yeah. These reactions use ATP and NADPH from the light reactions from the light dependent states. And the regenerations of the ribulose 5-phosphates is some of the triose phosphates. 5 out of 6 molecules is used for the regeneration of the ribulose 5-phosphates during the carbon dioxide fixations. And the process is in need of ATP. The rest of the triose phosphates is used to form 6 carbon sugars. 6 carbon sugars, and creates the sugar storage. Creates starch as a
storage of energy.
Storage of energy. Yeah.
All right, good. That's a lot. Now, I trust that number 1 is super easy for you. So organelles, structure and functions, super easy. Should be super easy for you actually.
Yeah.
So I'll pass. Now let's talk about organic chemistry, shall we?
Yes. Okay.
Oh, by the way, did you watch my videos on my science channel?
I did watch through all of it once.
Okay, good. So you did watch the video that I made about glycolysis. You did, right?
Oh no, I only watch chemistry.
Okay.
Yeah.
you will need those video in glycolysis during first year medical chemistry. Một
Okay.
mắt kem á, trong năm nhất nó chắc chắn sẽ cần đó.
Okay.
Right. Now, so organic chemistry. Can you describe the covalent bonds of carbons, multiple covalent bonds in carbon compounds, like how they are formed? Tại sao phải tạo được những liên kết 2, liên kết 3 gì đó? So basically, you have to talk about like the electron configurations and the formation of pi bonds and sigma bonds, you know, overlapping something, same fold. What else? Okay, just talk about it, please.
Can you repeat the question?
Covalent bonds of carbon, multiple covalent bonds in carbon compounds is the first topic you can see in organic chemistry here.
Covalent bonds. Covalent bonds. Carbon form four
bonds, yeah.
Yeah.
Because why? It has 4 lone pairs.
Lone pairs. They can have 4 covalent bonds.
4 lone pairs of electrons.
Yeah. Atoms. Create bonds. Covalent bonds to others. Atoms. The covalent bonds process of sharing electrons between the 2 atoms to form covalent bonds. And it can be single bond, double bond, or triple bonds. The single bonds is the result of overlapping 2 s orbitals, which the single bond contains 1 sigma bond, which is the overlap of 2 s orbitals. The double bonds contain 1 single bonds and 1 sigma bonds and 1 pi bonds. The sigma bonds is overlaps of the s orbitals, while the pi bonds is the overlaps of p orbitals, which overlaps sideways.
Overlap sideways, basically they're not real bonds.
Yeah.
Pi bonds are basically sigma bonds are way more stronger than pi bond.
Yeah.
Because basically sigma bonds are real bonds. The pi bonds are basically just overlapping of the electrons of the pi orbitals, or p orbitals, sorry. However, in double bond and triple bonds, you have both pi and sigma bonds. So double bonds and triple bonds are still closer and stronger than single bonds because of that. Now, so let's talk about aromatic and heteroaromatic compounds. We have phenol, we have aniline, we have salicylic acid, something like that. We have purine, pyrimidine, something like that, of course. Okay. What can you say about alcohols, phenols, and ethers?
Alcohols and what?
Phenols, alcohols, and ethers.
Ether.
Ethers.
They both have
Both là dành cho cả hai. all dành tất cả.
They all have.
They all have.
They all have alcohols and phenols both have hydroxyl
Hydroxyl group,
groups,
you mean?
and heat and ethers substitutes the H atoms with another's AQ groups. AQ groups, yeah.
Then you just need to write them down, and the reactions of the alcohols, phenol and ethers. Can you describe the reaction?
Reactions of atoms of alcohols.
We have what calls alcohol is in the most reduced form, nó là cái dạng bị khử nhiều nhất, the most reduced form.
Yep.
Cho nên là nó có thể là lên đến aldehyde, là oxidize lên aldehyde, hoặc là oxidize lên cùng lắm là acid. Và nếu oxidize thêm lần nữa nó quất luôn, nó mất luôn cái CO2 ra luôn, nó thành alkanes. Không, mà thực sự là mấy cái acid đó, em đốt một thời gian tự nhiên nó sẽ mất cái CO2 ra luôn. Basically acids, organic acids, COOH, if you burn it, after a while the CO2 escaped, and then what you have left is alkanes. So talk about the reduction of the oxidation of alcohols, can you? Not the oxidation, no. Oxidation of alcohols. It becomes phenol after it's oxidized. And you need to talk about what oxidizing agent you can use. Normally, cái oxidizing agent mình hay sử dụng nó là cái gì? Những chất nào? Ví dụ potassium permanganate chẳng hạn, KMnO4. Potassium permanganate, or you can say potassium dichromate, or you can say potassium chromate. These are all strong oxidizing agents that you can use. And so first, alcohol will become aldehyde, and then if you oxidize it further, it becomes acids.
Carboxylic acid.
Carboxylic acid, very good. Carboxylic acids.
And also secondary alcohols oxidize into ketones.
Ketones, very good. Primary alcohol will become aldehyde and then carboxylic acid later. Secondary alcohols will become ketones, but then you cannot further oxidize it, right?
Yes.
Không có oxidize được thêm nữa, thì đến ketone thôi. How about tertiary alcohol?
It doesn't oxidize under.
Tertiary alcohol doesn't oxidize further.
Yeah.
It's just alcohol. It doesn't. Okay. How about phenols? What reaction can you expect from phenol? No, acid nhẹ. It's actually phenol is like a weak acid.
Reaction can be reacting with a base to produce salt.
True, organic salt.
Yep, salt.
So basically, phenols can react with alcohol, isn't it?
Yeah.
To produce organic salt.
So
it can also produce. It can also react with amine compounds. Chỉ có amines, có N, nitrogens, amines compounds. because it's weak acids, of course, will react with anything basic. What else? Phản ứng cộng với lại chloride là chlorination reaction cộng với Cl2, nó quánh với octo, para, meta gì đó, you remember that? Like chlorination reaction, and the chlorine will go to octo, meta, or para depending on like the substituent on the phenol ring. Tùy thuộc vào cái substituent, tùy thuộc vào cái nhóm thế ở trên cái dòng đó, nó sẽ quyết định được là một cái substitution reaction nó sẽ đánh vào đâu, octo, meta hay para, nhóm đẩy, nhóm hút. It depends on whether the substituent is an electron donating or electron accepting group, you know. Have you heard about it? Have you learned it?
Donating.
Cái organic chemistry em cần phải học cho thật kỹ nha, cần phải coi sách học cho thật kỹ nha. Phần này yếu nhất luôn á. This part is like the weakest part that you have. We need that. We remember we need chemistry and biology. Okay. Now I think it's the end of the lesson. Now I'm so tired. Now it's already 2 hours.
Okay. Thank you for
Okay,
teaching.
I'll see you. If you want to have another lesson, feel free.
Okay.
Okay, I'll see you then.
Okay, I will see you.
See you then. Good luck.
Yeah. Thank you.
Let me know the result, okay?
Okay.
Okay, that's the end of my videos. Please like, share this video with your friends, subscribe to my YouTube channel, and press on the bell button next to the subscribe button so you can get notified about my new uploads, okay? You can also press follow my Facebook, but please don't add me on Facebook as friends, okay? Cảm ơn các bạn đã theo dõi nha. Các bạn hãy nhấn vào like, chia sẻ video này bạn bè các bạn. Nhấn vào nút đăng ký kênh YouTube mình và nút cái chuông kế bên cạnh để đăng ký kênh đó, để mà các bạn có thể được thông báo về những video mới nha. Các bạn cũng có thể nhấn vào nút theo dõi Facebook mình, nhưng nhớ đừng kết bạn Facebook mình nha. Thank you and see you. Bye.